Good afternoon, y'all. My name is Emma Vencer. I was fortunate enough to be hosted by the VR Labs this summer to work on a summer internship, and I examined plastic degrading bacteria. So to put things bluntly, we are in a crisis when it comes to plastic. In the United States alone, in 2016, we produced about 442 million metric tons of plastic in plastic waste. And once this plastic is produced, only about 9.3% is recycled. The rest ends up in landfills, either domestically or internationally. And once they're in these landfills, they degrade into smaller and smaller pieces of microplastics, which gets into our food, it gets into our water, and it gets into our environment. To put things in perspective, 442 million metric tons of plastic equals about 5,566 Olympic-sized swimming pools, 2,679 cargo ships, or 1,210 Empire State Buildings. It's a lot. Over my summer research project, I examined a couple types of plastic. The first type was plastic that's based in petroleums. So this is going to be anything that's marked 1 through 6 on your recycling index. It's your polyethylenes, your polypropylenes, your water bottles, your plastic cutlery, your takeout containers. Most of this falls under petroleum-based. It's really what's the most commonly used. And the thing that's significant about it is that it doesn't really degrade naturally in the way that we're going to look at some of the other plastics. It just breaks into smaller and smaller pieces. The next we have is plastic that's based in biomaterials, so stuff that's like from corn starches. It's marked as number 7 on the plastic recycling index. Typically, it's marketed as like your compostable plastics. This isn't necessarily the case. It requires large industrial facilities in order to compost, and that's really tricky in order to pull off in countries that don't have strong recycling infrastructure. And then the last thing that we have is plastic that's produced by microbes. This is going to be like your plastic straws that kind of start to grate in your mouth. That's the main use of them right now. Microbes use them in order to store carbon for them when they are in low nitrogen environments. They're significant because of this their structure. They're very similar to some sorts of petroleum-based plastics. By studying microbially produced plastics, specifically PHBV, we can hopefully use them in order to help mitigate the recycling issues that we are having with other sources of plastic. And so that kind of leads us to our research question is how do we isolate plastic degrading soil bacteria? And we're specifically looking at soil bacteria due to the sheer diversity of bacteria in the soil. And we're specifically looking at how to isolate these bacteria because while we know they exist, we need to know specifically like what strands of bacteria that we're looking for and how they work, and just to further study these mechanisms that they do. So in order to do this, I started with a serial dilution. So that's when you take a biomass sample of bacteria and you dilute it by a factor of 10, and then you plate those dilutions onto Petri dishes like this one. We used Petri dishes with R2A agar, and then over top of that we layered it with R2A plus PHBV, which is the microbially produced plastic that we're examining throughout this experiment. And it was powderized and like kind of just layered on top of it like a cake. Then we took these plates and with that were grown with the serial dilutions, and we plucked a colony off of that onto a new plate that we streaked onto it. And the point of the streak plate and the serial dilution is to hopefully get singular isolated colonies that are hopefully all genetic clones of each other. So we did this for about 3 to 4 generations. So in theory, we should have all of the same genetic material over time. And then this resulted in our plates. So as you can see here, right here, this like gray background material is going to be the plastic, and then this green mass is the bacteria. And you can see in this white spaces in between those two, that's actually places that was formerly plastic, but has since been degraded by the bacteria, showing that they have been taking in the plastic, using it for carbon, and then basically composting it, not composting, but using it for their own cellular processes. And then after we had all of our bacteria grown, we transferred it, we extracted the DNA using this QIAGEN kit, which resulted in a DNA sequence for us to read. And then we were able to identify the species of this bacteria. And these were our results of the identification results. In total, we had 5 isolates. We had 5 isolates, each with 3 different kinds of species identified. We had a Lactinobacillus, we had a Streptomyces, and then we had one isolate that we really didn't know what it was. Due to the nature of the sequence that we got back, we knew that they could have been 2 possible species, but we couldn't really tell which one. So all we can say is it was specifically from this family, and that's about all we can tell. And then these were our plates, our series of generations over time. And we were sure to make sure that with each generation that they each displayed plastic degradation at some step, which is significant because as we went on, we had some isolate candidates that didn't necessarily degrade throughout those 4 generations. At some point, they stopped degrading plastic, and that's something that we'd like to examine in the future, is like, why exactly do these bacteria stop degrading plastic after being transferred onto a new plate? We speculated that it could be something to do with competition on the plates, and that these bacteria will only degrade plastic when they're in competition with others, but we don't really know. Additionally, we would like to look at different organisms that can degrade PHBV. Specifically, we tried to look for fungi with PGA, which is potato glucose agar. It's like the bacteria food R2A, but it's more specifically targeted towards fungi. Nothing really came from our examples, but we know that it is a possible thing that could happen. Additionally, we tried to experiment with other types of plastic. So instead of the PHBV, which is what we used for the previous tests, we examined with PMMA, which is a petroleum-based acrylic plastic. And we created a gradient of different concentrations of PHBV and PMMA to hopefully get the bacteria that we have plated here to grow down the concentration gradient and see if they can degrade the PMMA up here. Unfortunately, these bacteria weren't necessarily the most motile, so they just kind of stuck up here. They didn't travel down the concentration gradient, so we don't really know if they have the ability to degrade PMMA. In conclusion, we have so much plastic that we need to find a way to get rid of in one way or another. One way that we can use to mitigate this problem is through the use of bacteria. And we approach this problem through isolating 5 new strands of plastic degrading bacteria to hopefully study them further and see how they can help us mitigate this plastic pollution issue. Thank you for your time. I wanted to take a couple minutes to thank both the RECCS team, the Fiera Lab for hosting me, and then also my mentors, Andrew Blakely and Nicholas Dargone. They were really wonderful, and thank you so much for your time.